I'm Peter Capainolo, and I'm here at the American Museum of Natural History. I work in the bird department, and the Museum of Natural History's bird collection numbers about 1,000,000 specimens. It's wide world in scope, and people come here from all over the place to conduct studies on birds. And the main type of specimen that we have is something called a study skin. And these are examples of study skins. These birds are common grackles. It's a New World blackbird that I've been interested in for quite some time now. And this is an old specimen. It was collected and prepared in 1888. And as you can see, the skin was removed from the bird and it was stuffed up with cotton. It's got cotton in the eyes, but many of the bones were retained in this skin. It's an old method of preparation, so the entire skull of the bird is in this specimen, and the beak is attached to the skull. The wing bones are still in the wings, and they're tied together to keep the wings together in the final prep. And most of the leg bones are still in there as well. So it gives the bird some support. The only problem, of course, is that back then you prepared a study skin and that was basically it. You didn't get any other information from the skeleton because it was discarded. A lot of the bones were broken off. And of course, back in 1888, nobody knew anything about DNA. And so no one took any tissue for DNA analysis. This is also a common grackle study skin. It's a more modern one. It was collected and prepared recently within the last couple of years. And the difference is that there's a lot more scientific information content in this specimen. The reason being that most of the bird's skeleton has been removed. There is no skull in here. The skull has been cut off at the base of the bill, so the bill is still on the skin, but there's no skull. Most of the wing bones have been removed. Only the hand bones have been left in the end of the bird's forearm. And only the lower leg bones are retained here. The long bones up at the top here, the femur, the knee, the tibiotarsus of the bird, those have all been prepared as skeletal material. In addition to that, a piece of muscle tissue has been taken and put in a -80 freezer, and that material can be used later on to extract DNA and look at the molecular structure of these DNA molecules. So with a specimen like this, you get the external morphology, a piece of DNA, and almost an entire skeleton. This is a specimen of a common grackle where the skin has been removed and discarded and only a skeleton prep has been made. But then you lose all of the important measurements that you can get from the cranium and from some of these long bones. So the goal here in using a new technique is to be able to get a lot more information from this bird with regard to measurements and regard to molecular material as well. So here we have a fresh specimen of a common grackle. Bird was collected in Kansas this spring, and I'm going to prepare it as a study skin in the using the modern method. The first thing you do is you inspect the specimen, you look at the wings, and you look at the tail, and you want to determine whether the bird is molting, if all the feathers are there, and some other aspects of the specimen. It looks pretty good. And of course, I'm always taking notes. I've already taken down initial information for this specimen. Date and location is very important. I indicated that there's no molt in my notes, and I weighed the bird, and it weighs 103.8 grams. Now, there is an interesting characteristic of these grackles, and that is they have, like all birds, something called a third eyelid, or a nictitating membrane is the technical term. So here's the bird's eye. You can see the iris. And then these are lower eyelid and upper eyelid. But in the anterior portion of the eye is this translucent nictitating membrane. I'm going to tease it out. And birds are able to move this nictitating membrane across the eye. And that'll moisten the eye and it'll keep out debris. And what's interesting about grackles is that they have a very elaborate courtship display. The males will sing and puff up their feathers in front of a female, and they'll flick their nictitating membranes across their eye. And if you look closely, you can see that there's a bluish color to this nictitating membrane. And we know that birds can see into the UV spectrum. And there's some thought that maybe when this nictitating membrane comes across the eye, that it kind of flashes differently to the females. And they are attracted to this part of the courtship display. So what I've been doing is harvesting these nictitating membranes. So the first thing I'm going to do before I prepare the study skin is I'm going to harvest those nictitating membranes. And it's fairly simple. Just take a forceps and draw it across and dissect it out. And I've been freezing the nictitating membrane of adult male grackles, adult female grackles, juvenile grackles, nestling grackles, fledgling grackles, grackles from different parts of their range in North America. It's a widespread bird. And at some point I hope to do some studies to determine whether this nictitating membrane is similar in grackles from one part of the range and another part of the range, and if it's different between males and females and young birds and adult birds. So there's a lot of information we can get from this one specimen. This one's in there a little bit, tucked in a little. Sometimes it dries after the specimen's been collected. And in my notes I will indicate that the nictitans has been harvested from this specimen. Or is that the one I already did? I don't think so. Okay. There it is. Third eyelid. It's actually a modified tear gland. Put that in a little weighing paper in a glassine envelope, and that'll be frozen. So this is a fresh specimen of a common grackle, and I'm going to prepare it as a museum study skin using this modern method where we keep a lot of the skeleton and the skin and we get a DNA sample. First thing I'm going to do is put a little bit of cotton in the throat to prevent fluids from leaking out as we skin the bird. So now I'm going to begin to prepare the study skin, and I'm using a little bit of very fine wood flour. When you start to skin the bird, some of the connective tissue against the muscles is kind of sticky, so I'm going to make a very shallow, short incision in the bird's skin, very thin, and just push it to the side. And I hope this doesn't obscure what I'm doing too much, but we use that to just keep the skin somewhat dry as we remove it. So I'm going to use the blunt end of my forceps to push the bird's skin away from its body, its musculature. You don't want to pull too much, and you don't want to use sharp-edged tools too often. You want to try not to make any holes in the skin. And so far I haven't done anything that isn't done in a standard skin. Nothing radical or new about this part here. But I have to disarticulate the legs from the bird's body. So here's the bird's knee right here, and I'm just going to push it up a little bit. And old preparators back in the old days, would just cut that bone in half and leave a portion of it in the skin. I want to preserve the entire bones for skeletal analysis. So instead of just cutting through, I'm going to disarticulate the knee joint. I just use my scissors very gently, and I just push the joints apart. Don't want to cut too much. Just push them apart. You can feel it give. And then I pull up on the tibiotarsus and skin it a little further down until I feel the ankle joint. I'm just going to gently twist that ankle joint around a little bit so that it starts to loosen up. And you can see this entire bone now is out of the skin, and we'll deal with that a little later on. So the only bone that's left in the leg is this lower leg bone here, the tarsometatarsus. Do the same thing on the other side, and that's usually not done in a standard study skin. Usually you just cut through the bone. Don't want to catch any feathers there. Knee. Now sometimes if a bone is damaged or broken because of the bird having been struck by a vehicle or hit a window and salvaged or damaged in some way, as long as you have one side, you have at least one measurement. Articulate that completely. So now I have two tibiotarsi bones. And as you'll see later, on the carcass, we have the femurs. Now the next thing we want to do is disarticulate the tail from the body. Birds have a tiny little group of vertebrae at the end of their spinal column, in the back of their pelvis, and those bones have to be snipped in order to get the bird's skin off. So we can't salvage those bones in this type of skeleton preparation. So I'm down near the abdomen. This is where the intestinal tract is. I've always got a little piece of cotton handy, because when you cut through there, you'll get some fluids leaking from the intestines usually. So I snip through that material and put some cotton in there, a little bit of desiccant, and what I'm going to do now is carefully pinch the tail away from the carcass. Some intestinal material comes out usually, and you don't want to get it all over the place and soil the feathers and make a mess. You can clean a specimen, you can wash it and dry it, but it takes an awful lot of extra work. So you try to avoid soiling the plumage as much as you can. And some people consider this the most difficult part of skinning a bird, getting this tail off. If you cut too close to the tail feathers at their root, then the tail feathers can come out. So you do have to be a bit careful here. And it's a little messy, so I use a lot of desiccant. At some point I'll be able to pinch that off and start skinning down the back of the bird like this. In a second you'll be able to see where we are. Hold the bird up like this. I'm skinning down the back of the bird and around the front, just peeling the skin down over the carcass. And you can get a sense of what's going on here. Here's the musculature. Inside are all the organs. These are the femora, the femur. And we disarticulated the tibiotarsus from the femur and from the ankle. So retaining these bones on the carcass for analysis. We can measure them and compare populations of grackles from different areas. The next thing we're going to encounter are the wings. So just a little bit of desiccant, it's a little sticky. So what you can see here is wing and the other wing and the neck in the center. Now in an old style prep, I would just cut through these wing bones, the humeri. But I want to keep the humerus, the upper arm, and I want to keep the lower arm, the radius and the ulna, and measure those. So instead of cutting through it, I'm going to skin down over the bird's elbow, down its forearm, just kind of push a little bit, and you can see there I've got the humerus and the radius and the ulna. Now this is the bird's wrist here. And a bird's hand, all of the bones are fused. So I can't remove those on a preparation like this. So I'm going to gently move that joint around. Get it a little loose, and then cut through the tendon. So that now I have most of the bird's arm. The wrist is here and the hand would be here. The hand is left in the skin. Do the same thing on the other side. We're moving down towards the bird's skull. So again, this is usually not done with a standard study skin prep. Down to the wrist. Move it around a little bit, hear it crunching a bit. I can see the distal part of the forearm. Snip through the tendon. And now get an idea of what's been done so far. This is the bird's skin. This is the body, and we're going to continue to skin down towards the skull. Now the skull is a very important part of the skeleton to measure when you're doing comparative studies. And in a typical study skin prep, you would cut the back of the skull off and retain it in the skin. But I want most of the skull. I want to be able to take some very important measurements of the skull. So I'll prepare this a little differently. First thing you have to do once you get over the back of the skull, the skin's here, the skull's here, is deal with the ears. Birds have ears. They don't have earlobes. They don't have any external structures to the ear. It's just a little hole in the head, and the skin goes down into that hole. So on a small bird like this, you can just grab the skin that goes down into the ear canal and pinch it and bring it up. So here's a hole in the skin where the ear is, and this is the opening to the skull that goes into the brain. So that's one ear that's been removed, or taken out, or skin has been taken over. Do the same thing on the other side, and continue to push this skin down towards the base of the bill. So the bird's inside out. The next thing we'll encounter are the eyes. And there's a little tube of tissue that you have to cut through to get to the eye. Very thin piece of material. Using the tip of the scissor, I'm skinning over the eye. You don't want to cut the eyelid, and you don't want to pierce the eyeball. Not the end of the world if you do, but the eye contains fluids that can stain the feathers, and then you have to wash the skin, and it's a lot of extra work. If you're collecting specimens in the field and you don't have refrigeration, you have to work fast. And you don't want to rush this part because it's more difficult to wash birds in the field than it is in a lab with a sink and different drying agents and so on. Same thing on this side. I'm just going to make one little slice through that tissue over the eye, and then do the rest with the scissor. I have a little more control with the scissor. And I'll use these fine forceps to finish. And my goal here is to skin all the way down to the base of the bill. So in a traditional study skin, what I would do at this point is remove the eyes and cut the back of the skull off here, freeing the entire body. Then I would remove the brain from the back of the skull and clean the skull up, put cotton in the eye orbits, and then turn that skin back right side out. But because I want this skull, I'm going to do things differently. First thing I'm going to do is remove or loosen the hyoid bone in the throat. Here's this piece of cotton that we put in the bird, and this looks like a piece of corn that bird had eaten. And I'm going to use a scissor that's a little more robust, and I'm going to cut the skull off at the base of the bill, fully freeing the carcass from the skin. So now I have most of the skull that I can measure, and I can still measure the bill of the bird that's retained on the skin. So I'm just going to clean this skin up a little bit, take off some of the connective tissue and fat that's clinging to it. Small bird like this doesn't have too much material to worry about. If it's a bird that's got a lot of fat on it, you do have to remove that fat or else it'll decompose and spoil the skin. I'm going to gently turn the skin right side out, push the beak up through the skin, and play with that skin a little bit to get the feathers looking fairly normal. Brush it a little bit, shake it somewhat. And then the last thing I need to do on this skin before we lay it aside and turn our attention to the carcass is remove a little piece of flesh at the base of the tail. Got a little meat left at the base of the tail, and there's what's called the uropygial gland, the dorsal part of the tail. This contains some oils that the bird uses to keep its feathers in shape. The bird will dip its beak into this gland and then run that beak over its feathers to waterproof the feathers and to repair the feathers. Just going to remove this little piece of caudal bone that's in there, and unfortunately it's too damaged to do anything with. You have to sacrifice a little bit of that bone. Just going to get a little bit of flesh off of here. Get that uropygial gland off. You don't want to cut too much because there is a chance you could ruin the base of the feathers and they could come out of the skin. Get enough off there to clean it up a little bit. So it's actually good to leave a little bit of flesh there so that the feathers are embedded in it. It'll just dry up. Okay. So that bird is essentially all skinned out. Now the second step will be to stuff it full of cotton, but before we do that, we'll prepare the carcass so that it can be turned into a complete disarticulated skeleton. So I'm going to just clean up here a little bit. Turn my attention to the carcass. Move this tray so you can see it. The first thing I'm going to do is get a little sample of muscle that we can freeze and then later on get some DNA out of it. So I'm just going to score some of the breast muscle with the scalpel. Couple directions. Got a tissue tube with my specimen number on it and a number that goes with the tube. A barcode as well. And I'm going to just put some of that muscle tissue into that tube. And that will go into our cryogenics lab, be stored at -80 for use later on. Now the way a skeleton is prepared is we remove most of the musculature and leave a little bit of flesh on it, and we have a colony of dermestid beetles. The larvae of those beetles are carnivorous, and after we remove most of the muscles and dry the specimen out, the beetles will do the rest. They will eat all of the flesh that's remaining on this bird, and we'll get the skeleton back as a little pile of disarticulated bones. But I do have to prepare this a little bit, and I also want to check the internal organs of this specimen. Male and female grackles look somewhat similar, and birds have internal sexual organs. So the testes are inside the abdominal cavity in males, the ovary is inside the abdominal cavity in females, and in order to be able to determine the sex for sure, we need to open up and look at those structures. We can also see the size of those structures, and that gives us some indication of the breeding condition of the bird. If they're enlarged, it means that the bird is in full breeding condition. It's got eggs or young in the area. If they have recrudesced, if they've gotten really tiny, it means that the birds are finished breeding for the year and they're ready to migrate. So the first thing I'm going to do is I'm going to just strip the meat off of these long bones that we got from the legs, and it's not difficult. Just pull it all off like that. And you don't have to take every scrap of meat off because the bugs want to be able to eat something. We affectionately call our domestic beetles bugs. Then I'm going to just remove the breast meat pretty much all in one piece. Leave a little bit on there. Do both sides. And the goal here is not to score any bones or to accidentally pull any bones off. Disarticulate the trachea. Pull that up a little bit. I'm going to keep the tongue with the specimen. I'm going to take a little bit of flesh off of the upper arms. Take a little bit of flesh off of the femur of the hind limbs. And the goal here is to take off enough flesh that you can dry this out. The beetles like to eat the meat after it's been sort of air dried. So that's probably enough flesh taken off. What we want to do now is look inside the body cavity at the condition of the gonads. So remove this plug of cotton. Make a very short incision in the upper part of the outside of the abdomen. Going to dissect out the liver and the intestines, the gizzard, the digestive tract. that material. And you can open the gizzard up and see what the bird's been eating. For some studies we do that. And this bird is a female. It might be a little difficult to see, but inside the body cavity, up on the left side, on top of one of the kidneys, I'll remove it, it's the ovary. That's the ovary, and it's not very developed. This bird was collected in early June and may have already laid eggs and raised all the young of the year. And so this ovary is in the process of shrinking up for the fall and the winter. If it were a developed ovary, you would see each individual egg cell very, very large. What you can see here are what look like a little bunch of grapes when I press on it. Those are all egg cells. So I'm going to finish preparing the skeleton, at least the pre-prep for the skeleton, by removing some of the internal organs, the kidneys, heart, the heart, and then I'm going to insert these leg bones that we disarticulated inside the body cavity. I've got a label with my specimen number on it on a string, and I'm going to wrap this carcass up so that the forelimbs and the hind limbs aren't sticking out. I'm going to wrap it sort of gently. And it'll be put aside to dry, and then when it's dry enough, in a couple of days, it'll be sent over to the domestic beetle colony, and the beetles will do the rest. The next thing we want to do is finish preparing the skin. I'll clean my tools here a little bit. Make sure my fingers aren't too bloody. Come back to our empty skin, lay it out, stretch it a little bit. So this is interesting. We know we have a female common grackle for sure, collected in Kansas in June. So it's very useful for studies of biogeography, demographics, systematics. This specimen will be useful for several kinds of biological studies. Now, one of the things that is done in this newer prep type is I sew the feather tracks together on the wings to keep the wings together in a finished study skin. In the old time preparation, these bones would still be in here, and you could just tie those wing bones together. So what I'm going to do is just bring up a little region of the wrist that was left in the skin, and instead of tying any bones together, I'm going to stitch these together with a little bit of thread. So there's a feather track here. You can see it on the skin running down there. I'm going to just go through that feather track with the thread. Same thing on the other side. these together. So we have some way of giving some support to the wings in the finished study skin. Now that the wings are tied, I'm going to take a small dowel. It's not very thick. It's pointed on one end. And I'm going to insert that into the skin of the throat and down into the skin of the body cavity above where I tied the wings. This end will go into the cloaca of the skin and protrude out like that. In an old-time study skin, you would insert this pointy end of the dowel into the base of the skull. But I'm going to insert it into the palate, in the upper part of the bill. And that is going to serve as a replacement for the bird's spinal column. So all study skins have some kind of dowel or piece of wood or wire or something in their body like that. Now I want to give this bird some cotton eyes. We like to use this cotton that has long fibers. You could use anything, but this long fiber cotton running in one direction is very good for making study skins because it holds its shape. So I'm going to just make a little ball with the cotton. Approximately the same size as the bird's natural eye. Birds have very, very large eyes. Most of their brain is eye. And I'm going to run this up the neck skin and tuck it in behind the dowel on one side and just let it stick out a little bit into the eye. If the skull was in there, this would be much easier to do because you'd have the bony eye socket to insert that cotton in. In this case, we've removed that part, so it takes a little bit more effort to keep that cotton in there. Make another eye. It doesn't seem like this kind of work will ever be automated. It takes an actual human being to prepare the specimens like this. And so it's a tradition in field ornithology to sort of pass down the techniques from teachers to students. Now I've got cotton inserted into each eye. Because we removed the skull, I need to make a little faux skull out of cotton so that the head has some shape to it. Just take a larger piece of cotton and make a larger ball out of it. Nice and wrapped so that it's tight. quiscula replacement up behind the dowel into the neck skin and up into the back of the head. Kind of holds the eyes in. And because it's cotton, you can shape it a little bit. The bird doesn't have to look perfectly alive, it just has to be able to lie flat in a specimen tray and be useful for measurements. Another group of people who really benefit from study skins are artists, people who paint birds for field guides, use specimen collections all the time to get shape and color for their paintings. So we've got the head finished. All that's left to do now is to make a cotton body and then sew it up. So to make the body, we'll take a long strip of this long fiber cotton, like that. A couple of other pieces that I'll need for the body. I'll separate these out a little bit. Now you may not be able to see what I'm going to do in the video because I have to roll this cotton. So I'm going to just roll this on my knee so that you can see it. Just going to make a nice tight long rope. You don't want to do this with your dress pants. Jeans are best. So I've got this fairly strong cotton rope, and I'm going to roll it up at one end. And I'm going to take separate pieces and I'm going to start binding them to the middle piece and start to make a little artificial body for the specimen. Now some people fill their birds up with a lot of cotton, make them nice and fluffy. Other people use less and the bird looks a little more skinny. It's really just a personal preference. If I wanted to get this body exactly right, I could take measurements of the carcass that we removed and get it exactly to those measurements. But I'm going to just eyeball it. It's fine. And you just want it to be sort of teardrop shape. And once you've done a couple of thousand of these, you get pretty quick. It could probably be a little bigger. Just a little bit more. And it looks somewhat smaller than the carcass sometimes, but it will do. Let me shape it a little bit. So that's the body that we're going to put inside our skin. So grab it with the forceps at the end. I'm going to feed it up the neck. Don't want to catch any feathers or skin, and I'm going to have it protrude out of the mouth. Get a hold of it there. I'm going to pull it through, bringing the skin over it. And there's nothing really unusual about this part of finishing the study skin. This is pretty typical procedure, regardless of what you do with the bones. So bring that skin over it, fill the bird up. Make sure the skin isn't catching up on the cotton. A couple of things you want in a study skin, that is the neck of the bird should be fairly short, unless it's a bird with a long neck like a heron or a crane or something like that. You might want to extend the neck to show the characteristics. The other thing you want when we're finished is you want this bill to be sort of angled back. You don't want it sticking up at a right angle, because there's trays in the cabinets where the birds are kept. And if they were all at a right angle, they would be scraping against the tray above them. And you want the back to be sort of flat so that they don't roll in the tray. You want to be able to store a large series of skins in the same trays. If you get a few birds over many years from different localities, then you want to be able to place those birds with each other based on locality, so that your series starts to be valuable scientifically. You can look at populations and not just individual birds. So I'm just shaping it a bit. Pick it up by the stick and play with the back of the bird a little bit. You're not making a taxidermic mount where it needs to look alive and perfect. Some people make beautiful skins. They're just, every feather is perfect and the birds look really good. There's some famous people like George Sutton, who used to make perfect skins. Every one of them was exactly the same in terms of the prep. So I've got that bird filled mostly. I'm going to do a couple more things. I'll take a little bit of cotton and make some strips and tuck them in on the sides to sort of fill out where the leg muscles were. So that bird is filled up pretty well. Take our needle and thread, close the bird up. You don't need a double thread, just like that. Start at the top center, pull that through, and the idea here is not to catch too many feathers, and you don't need a whole bunch of tight stitches. Five or six will do. Just in the skin, along the edge of the incision. last stitch goes in the flesh near this cloaca in the center. And the stick's protruding from. And I usually pull up on it a little bit to close that pouch at the bottom. And just draw it together. Tie it off at the bottom. Some people will leave the stick protruding out long like that and not handle the skin by the body. They'll use this stick as sort of a handle. I usually cut the stick short so that it's just coming out of the skin a little bit. Start to arrange the feathers a little bit. There's a couple things we have to do here. The first thing is tie the legs together. So we're going to put the left one over the right like that. And I've got another label prepared here for the skin. So this is my specimen number. And I've got the weight of the bird on here. It's not absolutely necessary as long as it's recorded in your catalog and in the museum database. And you don't wrap the string around both legs side to side. You go under the legs. So that they don't roll around on each other. Tight like that with the toes facing inward. Spread the tail a little bit. The cotton that's coming out of the mouth gets cut off close to the opening of the throat. And we use that cotton to fill up the throat a little bit. Remember to angle the bill back. I'm going to use some leftover thread and needle to sew the bill closed. And then when the specimen dries, we can remove that thread around the bill, and the bill will have dried closed. To bring this thread around a couple of times so that it closes tightly. You notice I put the needle through the bird's nostrils from one side to the other so that I could have something for the thread to sort of rest on. If you just put it around the bill, it'll slide off. So I'm going to make sure that the upper and lower mandible are in the right position. some of that extra thread. Be nice if the scissor was sharp. Be nice if both scissors were sharp. There you go. And now, to finish, I'm going to just tuck the wings in a little bit. They're a little bit loose because we don't have any bone in there like a traditional study skin. We've just sewn them together. So I'm going to just shape the bird with my hand a little bit, move the cotton around. The wings will dry in position, but I've got to make sure that they're in the proper place. Take these flank feathers of the bird and cover the edges of the wing. It's going to help keep them tucked in. Like that. The legs will get tucked up a little bit. Most of the rest of the preparation here is just primping. And that will sit in a cool, dry place for a couple of days. And once the skin has tightened up, remove the string around the bill. It will eventually get an American Museum of Natural History label that corresponds with the data that's in my field catalog. And as you can see, this Grackle specimen was prepared the same way. That's ready for measurements and to be used. This is the older specimen from 1888 that has the skull in it, has the wing bones in it, has most of the leg bones in it. So this old specimen has value in terms of what you can see on the feathers, the color, the shape of the bird to some extent. But these two birds have the skin, most of the skeleton, and tissue for DNA analysis. And so we increase the scientific information content by preparing them this way. That's basically the procedure.